Okay, folks, let's get started. Greetings. I hope you guys are ready for a big, exciting day. I am. I've warned you about this, so I'm actually streaming live on Facebook right now. So, yes. This is the first time I have ever used a selfie stick, so this is a new experience for me. I've only had one problem so far, and that was PowerPoint. And if there's one god-awful creation that was ever made by man, it is known as PowerPoint. So, but I think I've got it fixed, and we should be set. As all of you know, this is my last lecture, and this is a very big and emotional day for me. I'll say a little bit more about that as I get into a presentation I have later. First things I'll talk about today are actually biochemistry. And then I've got some other things to talk to you about. Before I talk about the biochemistry, I want to say that there is a review session that I'm going to hold. I was going to hold it here, and I decided not to mix things up. So the review session I have scheduled for the class is on Monday at 5 p.m. in ALS 4001. And as usual, I will videotape that. So that's the review session. Don't forget, we have the final exam in here on Thursday at 9:30. And some questions about the final exam. The final exam will be about 150 points, so it'll be about 1.5 times as long as the exams you've taken. You will have a full hour and 50 minutes to take it, so time usually is not a factor, shouldn't be a factor for you on the exam. The format of the exam will be exactly the same as the format of the first two exams that you've seen. May change a little bit in terms of percentages of points from each thing, but basically it should be the same format of the exams that you have seen. Okay. And I've mentioned before, I'll mention it briefly here, people say, what strategies should I use for studying for the exam, etc. And as I say, I'm the worst person to ask because everybody studies differently, but I'm a big fan of writing things down. I'm also a big fan of using the things that we've talked about previously as the sort of organizing outline for your study for the exam. So that's a good thing for you to do. And as always, if you have questions of me, please feel free to come by, questions of the TAs, etc. And don't freak out. I guess that's the last thing I would tell you. Yes, question? Can you go over the breakdown of how like what's from what? Okay, so breakdown of points. I try to make things proportional to what we have covered in the class, which means that we're going to have roughly 70% of the material from things we've already covered in class and about 30% for things material that we've covered since the last exam. Okay? If you haven't gotten your exams, you can still pick them up. They're available in the VB office in ALS, I can't even think, 2011. My mind isn't working today. Okay. Other questions? But yes, Hannah. The office will have the same hours next week. And if you can't pick up your exam when the office has their exam pickup hours, just come see me, and I'll be happy to do that with you. As I've said, my schedule's been a little bit crazy and funky this term, so if you want to make sure you catch me, send me an email, and I will definitely schedule something with you, okay? And I'm happy to do that, so happy to do that. As I said last time, I have really enjoyed this class a lot. You guys have been very interactive. I've had, I think, more visits to my office from this class than from any class I've had in about the past 15 years, so it's been very, very cool. So thank you guys for your interest and your participation in that. Well, I only have two minor things to talk about with respect to biochemistry. I did that on purpose because I wanted to spend some time talking about my last day and where you guys have fit in and where you've made a difference for me. The last things we've been talking about, of course, are nitrogen metabolism and how nitrogen is important, and more specifically, we've been talking over the last lecture about amino acid metabolism. And I show you this pathway not to give you another thing to memorize because I don't think that's beneficial. But I do think there are some general take-home messages with respect to amino acid metabolism that you should understand. The first of those was when I talked about the synthesis of amino acids, I broke them into 6 groups, one of which only had one member, and that was histidine. And I noted that the synthesis of amino acids was not in any grand scheme coordinated. With the families themselves were coordinated, but there weren't coordination between families to do that. The breakdown is sort of like that, although we do, with breakdown, think about the grouping of amino acid breakdown into 2 or 3 main groups, and these relate to the intermediates that they ultimately produce. So this is a big simplification, and I know that doesn't look very simple, but this is a big simplification of the overall process, and I see some students from last year's BB451 class who've decided to join, so happy to have them here too. But the simplification is that there's really 3 categories that you can see up here. Amino acids that break down in a manner that we call glycogenic, meaning that they're broken down to glycolysis, gluconeogenesis intermediates. Amino acids whose breakdown pathway is ketogenic, meaning that they break down into acetyl-CoA, and those that are both, which means that they make potentially intermediates in both of those pathways. I've colored these in red, green, and blue according to where they fit in, and then I've overlaid this onto the citric acid metabolism, citric acid cycle, which is, of course, a central metabolic pathway, and you can see how all of these interrelate. You see some appear in 2 places because there's various ways that some of the amino acids themselves can break down. I don't really have much more to say about them than that. To get to the point of being one of those intermediates is sometimes a very elaborate breakdown process, and that's more than I want to spend time going through here. But this takes us back to the very first thing that we learned in the course, and that was that the citric acid cycle was anaplerotic, and it had relationships with all kinds of other pathways. And I think at this point you can see those relationships with those pathways. So that's the bottom line of what I would tell you there. Amino acid metabolism is not trivial. From a health perspective, there are many diseases of amino acid metabolism. I've listed some of those there. Again, not to give you something to memorize, but to just illustrate to you the importance and the effects of genetic disease on various aspects of human health, as you can see here. So that's really what I want to finish up with respect to content in the course. And that's the last of the material that'll be there for the exam. So if you need to run or go do anything, there's nothing else I'm going to say today that you're responsible for. So feel free to do that. What I'm going to do with the rest of today is I've got a sort of a memories presentation that I'm going to give to you. That is very important to me. And we'll finish with some music, which to me is very important to do, as you guys all know. So we'll do that, and I hope that we'll have a lot of loud singers at the very end. Well, I want to start out by saying, before I actually start the presentation, that I am extraordinarily fortunate to have been able to do this for my career. The first big class that I taught was actually BB451, and that was back in about 1996, I believe, and I co-taught it with my PhD mentor, Dr. George Pearson. I got my PhD here at Oregon State with Dr. Pearson, and I was extraordinarily nervous with that, because the worst thing to do is to stand in front of your PhD mentor and look like an idiot. And I didn't want to look like an idiot, so I was very, very nervous, and I worked my tail off. And one good thing about being nervous is it pushes you to work harder, and I worked very hard that first time. To give you an idea, back in the very first BB451, we taught it in quarterly 1109. There was about 100 students in it, okay? And that seemed like a lot of people. We taught it with an overhead projector. We didn't have PowerPoint. We taught with an overhead projector, and I brought the innovation along. I was the first one to do this. I'll take credit for that. This wasn't much of an innovation, as you will see, but it seemed like at the time I was the first one to record my lectures and post them on the web. Not record video, record audio. And that was a big deal at the time. So people could go to the web and listen to the audio lectures and so forth. So we did that. As I say, we used an overhead projector. Started videotaping the lectures in 2001, and that was 17 years ago. And at that time, YouTube didn't exist, so we posted the videos on the OSU website, and OSU's bandwidth was so small that when you looked at this on your screen, the image was 1.5 inches wide. And everybody thought that was the coolest thing. Well, you couldn't see anything, and I think the reason they liked it was A, it was kind of a cool new gimmick, and B, they liked the audio, which they were already getting anyway. So over time, it's expanded, and of course, now we've got full screen video and so forth of that, as well as live Facebook here. Okay, so I want to acknowledge, in fact, I want to acknowledge Chris Matthews, who's sitting back in the back, who was instrumental in helping me to get to teach that very first class. I taught a couple classes before that, but 451 was the first big class that I taught, and he was instrumental in the whole thing. So thank you very much for that, Chris. Also, Dr. Gary Merrill is back there, a colleague who has also played a very, very important role in my career, and I'm very grateful to Gary and very happy to have him here, as well as everybody who's here. Okay, well, let me pull up some memories and show you a little bit of that. So let's see, that's coming later. Okay, so we will start this guy right here. Okay, so my last lecture, all right. This was a street, Indira and I took a vacation in Newfoundland, and if you ever get a chance to go to Newfoundland, go. It's an incredible place. Actually, no, I take it back. This was Nova Scotia, but Newfoundland is an incredible place, as is Nova Scotia. And we're walking by this park, and Indira says, "Oh my God, look at that." And so, of course, I had to go pose for a picture, and so I thought it was a good thing for my last lecture. I've done this sort of semi-chronologically, but it's not totally chronologically. This is one of my favorite sayings, and many of you have heard me say this as I've advised you over the years or as I've met with you in my class, and that is that life is what happens when you're busy making other plans. John Lennon, that's the lyric of a John Lennon song called Favorite Boy. What is it? Beautiful Boy, that's what it is. Yeah, I couldn't think of it. Sorry. My brain isn't working very well today, guys. So you say I can blame it on last day, it's that, right? So maybe it's a good thing I'm getting out of this. This was the house I grew up in. I grew up in a little tiny town of 200 people called Fowler, Illinois, which was about 15 miles away from Hannibal, Missouri. That was the closest thing that you might recognize. Not too far from the Mississippi River. I spent the first 17 years of my life in this house. It's a little tiny house. There were about 2.5 bedrooms for 4 kids and 2 adults. The thing on the end of the house got built on when I was about 13 years old, and so all of a sudden we had 2 real bedrooms to live in. To give you an idea about what that time was like, I was born in 1954. And all the way through the time I lived in Fowler, which is what this town is, the houses on either side of us had no indoor plumbing. So they had outhouses. They went out and did their business all months of the year. Our house had plumbing. We had plumbing, so we were fortunate. But they had no indoor plumbing. And I look back at that and I think, wow, you know, when I think about things are tough or whatever, and I think about going out and visiting those outhouses in January in Illinois, it's not a very pleasant thought. This was me and my siblings. I'm the tall, skinny one on the right in the back. On the left in the back is my sister Kathleen. In the front, on the right is my sister Erin. And watching today, I'm not sure if Erin's watching today. I know my brother Brian is watching today. Brian is on the lower right, and just so he gets a chance to see where he's at, there he gets to see it. So, happy to have him here and watching. I show this picture, a little embarrassed to show it actually. I posted this as my Facebook picture one time, and I got a comment that somebody said, "You used to be cute." I thought, hey, wow, I think I like that compliment. So I thought I'd put my cute picture up on my last day as my last lecture. This is before I started wearing glasses, and back when I still had some hair. This was the family. My father and mother you can see here, and of course I'm the guy in the back with the black glasses, exactly like my father. And by that point I'm probably about 13 or 14 years old. And it was a big deal to go to the photographer and get that taken. That was not done with the selfie stick. One of the most important things educationally for me, interestingly enough, was attending this high school, Unity High School in Mendon, Illinois. And the Unity High School chemistry class headed by Babette Suzanne is watching today, live on Facebook. And it was in the chemistry class in this high school where I went that I learned chemistry and I learned mathematics. The two teachers that I had there for chemistry and mathematics, who I will acknowledge later, Earl Carpenter and Dick Spohr, made things so clear and apparent to me that I never in my life, you'll think I'm making this up, but it's true, I never ever had to study for a math class again in my life. Dick Spohr was an amazing man. I got to see him at my high school reunion this past summer, and I told him what I'm telling you here, that he made it so easy for me. And he spent the first month of class teaching us, and you're going to say, what the hell? Teaching us what the equal sign really meant. I've never seen anybody teach math this way, and he did it, and it was remarkable. And because of that, as I said, I never had to study a math class again. Unity High, the town I grew up in, Fowler, was so small that it didn't have its own high school. So there were about 5 or 6 towns that went together to have this high school called Unity. And Unity, my graduating class in Unity High School had 72 kids, to give you an idea about how big it was. Wonderful place, wonderful people. And I am very fortunate to have had the chance to go there. A little tiny high school, you know, I talk to students that go to little schools, little places, and they think that they're at a disadvantage. And I have to say, I felt all along that I had the advantage because I got very personal attention and I got very excellent education as a result of that. So I hope some of you who have gone to smaller schools will recognize that, and I'm sure you probably recognize it by this point in your training. When I was a senior in high school, I went to move with my parents to Stillwater, Oklahoma, where this is very self-indulgent, I'm feeling a little embarrassed, where I graduated high school. Actually, I didn't graduate from Unity, I graduated from Stillwater High School. At Oklahoma State University, I was able to go to school because my parents lived in Stillwater, and I was very fortunate with that. In those days, which was back in the dark ages, undergrads didn't do research hardly at all with professors, and I didn't either. So I was a goofy undergrad bouncing around through 5 majors when I was an undergrad, believe it or not. And the degree I ended up with was zoology. It wasn't biochemistry or anything like that. I see some BB freshmen over here, so happy to have you guys here. When I graduated, I had the very good fortune of getting a job in biochemistry. And I got a job in biochemistry because I had taken as an elective a linear algebra class that I took as an elective because I liked math, and math was easy for, like I said. And the professor here, who's E.C. Nelson, who's in the back right of this photo, liked the fact that I'd taken a bunch of math, and he hired me to be a technician for him. So I got to work in this lab with EC, a bunch of people there I won't name, but on the lower left, Dr. Bruce Howley, who was a postdoc in the lab, is a very good friend of mine. Both Bruce and EC Nelson, lifelong friends. EC passed away in December, sad to see that, but had a wonderful relationship with him and learned a tremendous amount of stuff from him. The lab that I worked in did research on vitamin A, and so we were very interested at that time in the metabolism of vitamin A. What is it that happens to it after it's in the body? We know it's important for vision and so forth, but we were interested in the specific form of it known as retinoic acid. That was me with hair while I was a graduate student in Stillwater, Oklahoma. I got my bachelor's in zoology and then my master's in biochemistry from Oklahoma State University. That's me shortly after I arrived in Corvallis, when I was a graduate student here working on my PhD. And again, I still had hair, but you can see what graduate school does to you, right? So this is me as a postdoc. After I finished my PhD here at OSU, I had the good fortune of getting a postdoc at UCSD, working with Dr. Rick Fertel. And the lab was an incredible learning experience. Had a wonderful time. We worked on Dictyostelium, which are slime molds. And if you ever want to learn about slime molds, come see me sometime. I'll tell you some cool things about them. The boss had just gotten a new couch, and he was very proud of the couch, and so we decided it needed to be roughed up a little bit. And so we decided to do that. This was about half the people in the laboratory, and you can see that the center person, that's the center problem troublemaker there, was not me, but you can see who she was. And of course, many of you know my wife Indira, who's sitting in the center in the back, and she's got that look on her face like, damn it, I'm gonna really mess up this couch. Made wonderful friendships as a result of interaction with all of these people. And we had a lab of about 25 people, so it was a wonderful, wonderful learning time. This is my favorite photo of Indira and me. Indira has been absolutely essential for everything that I've ever been able to do, that has been teaching, that has been writing, that has been you name it. And she's been wonderful. We met as postdocs when we were in that last picture I showed you. This picture was taken for a promotional photo for our first open educational resource book called Biochemistry Free and Easy, and very happy to have that. This was taken by Gilda Cumming, by the way, who's a photographer in the Honors College, who does a wonderful job with her work. This is one of my favorite photos. Don't tell Indira I showed this photo. She probably will kill me, okay? But this is taken one of our favorite places on Earth, and this is from an island called Galiano Island, which is off the coast of British Columbia. And we go there about once a year and just kind of chill and relax and watch a bunch of stuff. I'm going to show some students today. I've had way more students than I can either show or acknowledge, and I apologize in advance that I'm not ignoring anybody and I'm not picking preferences, but these were ones that I happen to have some pictures of, and some people that I'll acknowledge in a bit that are also important as well. I showed this photo because it was one of the first big classes of biochem students that I interacted with in an advising capacity from day one. And so I worked with these students from day one as an advisor, and you can see there were an incredible group. Indira's in there as well. I didn't advise her, believe me, she advised me if anything happened. Okay, we had a lot of fun. One of the things that Indira and I started in the department was the BB Club, which is the Biochemistry Biophysics Club, now called the Biochemistry Club, that we worked to get student participation in, and we worked to generate funds for. So we decided one year that we would have a Pie the Professor in the Face contest, and we opened it up actually to BB 450 and 451 students. They had to bid how much they were going to pay to pie people in the face. And so I think I generated $300 or $400 for the club, and believe me, that was a lot of pie that came as a result of those efforts. You can see our current department head, Dr. Andy Karplus, also was one of the participants, so people participated for a very good cause. Two of the people in that last photo decided at Halloween in their senior year that they wanted to dress as Kevin and Indira, and they didn't tell us this, okay? So this was a really cool idea. So I had my own surprise, which I was going to do, and I'll show you that on the next photo. This wasn't Halloween night. They redressed up for this. But I dressed up as this Dracula character and didn't tell them I was doing it. Well, they didn't tell me they were doing this, and they were two TAs for me. So I came to my BB 450 class, I snuck away into this room, put on my Dracula costume and so forth. Meanwhile, they snuck away and did this, and then Tony, who's the one in the back left, came out dressed as me as if he's going to give the lecture before I got a chance to get out there. And of course, the class does this sort of double take, kind of like you guys are doing right now, right? They did an incredible job, as you can see, of looking like the two of us. That was my Dracula outfit. I had a terrible joke, and I'm going to tell it to you here because you guys know I like jokes. I had a terrible joke that I told that day. So if anybody gets the answer to this joke, I have prizes to give. So I'm going to give a prize to anybody who gets this punchline. What is Dracula's favorite sugar? Say what? What is Dracula's favorite sugar?
Blood sugar?
Not well. No. Blood sugar is a good guess. Is this a terrible pun? Oh, come on. Sucrose, right back there. All right. Come get your prize. A copy of the Metabolic Melodies. So, yes. Awesome. Okay, that's very cool. You tell a couple more jokes here. We'll liven the thing up a little bit. First thing everybody's happy about. Your reaction was very different than that of my class. When I told the class that joke dressed as this, I heard this collective, oh, you know. When you hear that, you know that you've actually made a good pun. Okay. I may have told this one in class as well. Another joke I made up. What do you call people who worship paper bags? I haven't told this one, or else people weren't paying attention, one or the other. What do you call people who worship paper bags? Over here, yes. Sacrilegious, you got it. Come get your prize. Had you heard it before, or did you figure it out? You figured it out? Good for you. Good for you. Okay, excellent, excellent. Okay, I've got 2 more prizes. I've got to figure out what to give them away for. I don't know. We'll think about that as we go. Not everything was teaching and fun and games, but everybody knows that I'm a giant Beatles fan. And so on one of our vacations to England, I got to go to this place. And for anybody who's old enough or enough of a Beatles fan to know, this was the famous Abbey Road crossing where John, Paul, George, and Ringo crossed and had their photo taken for the very famous Abbey Road album. And so it was a wonderful opportunity to go and participate in that. And I will tell you, this is really hard to do because if you ever go to this place, there's usually a line of people to get across it. So just being able to be the only one in there was really a challenge. So I was very fortunate to do that. Okay. We went to, on the same trip, we went to Dublin and discovered that there was a church there called St. Kevin's. We never found the church, which I think says something about Kevin. But we did find this grocery store, and I thought, well, I had to have a picture of that. Besides being a professor, I also perform weddings. I do. I perform weddings free for my students. So if you ever want to get married and you want me to marry you, let me know. If I'm around and I can do it, I would be happy to marry you. Okay? This was a wedding. McKenna Parker was the student on the left who had been in my class. And she asked me if I would, she had heard I married people, would I be willing to marry her? And I said, sure. And so I tied the knot for the 2 of these guys. And it was a lot of fun. I did one wedding of a daughter of a friend of mine whom I knew since she was an infant. She was the first one I married. And she said, would I be willing to marry her? And I said, well, I'm not licensed, but I could go do this. So for $50, I went out, I got the things necessary to get the permission to do it. And then she said something that I think was a mistake. And I said, what do you want me to do? And she said, surprise me. Keep an eye on the timer. Surprise me, right? So what did I do? Well, I wrote the service. They include all the vows and everything. And I wrote it in verse. So the whole thing was in verse. She didn't know this in advance. It's a true story. I wrote the wedding song, okay, that they marched to, which she knew that was coming, but she didn't know what it was. She didn't hear it till the day of her wedding. This is what a sport she is, okay? And then at that wedding, I went out and rented a costume, and I came out dressed as William Shakespeare doing this thing. And so you should have seen the reaction. I was very nervous because, again, oh my God, I'm going to ruin somebody's wedding. And I told her beforehand, I said, you know, I'll be happy to tell you what I'm going to do beforehand if you want to know. And she says, nope, I want to be surprised. So she was surprised. It worked out really wonderfully and had a great time, but it was a little nerve-wracking at the time. I'm also, a very good friend of mine is a man named Neil Gladstone, who is a musician here in Corvallis with incredible skills. And he writes really silly, funny songs. He's been my inspiration for many of the songs that I've written. And he created some songs that we performed at the Majestic Theater on 3 different occasions with a group of people that are called the Ginseng and Tonic Singers. And I guess the first person that could tell me which one up there is me will get a prize. Which one? In the blue. Rania got it. Can somebody pass that back to her or whatever? So, okay, you can climb over people. Excellent, Rania. Okay. Anyway, these were the Ginseng and Tonic Singers. We had a great time with them, and Neil Gladstone is a wonderful inspiration. A more formal singing was done by a group of young women known as Divine, who are the a cappella group, one of the a cappella groups here at OSU, and they performed the Metabolic Melodies about 1.5 years at LaSalle's Stewart. And I was very delighted to have them there to do that, and I was very proud actually to be here posing with them. I don't know why I put that one in there, but Dan Edge is a friend of mine here on the campus, and Benny Beaver, of course, is everybody's friend. Last year, I had, there they are, there's 2 people sitting in the front row who are in this picture. Last year, I had a group of students that called themselves Aher Nation, who studied together for the whole year. Really remarkable group of students, and this is everybody in that group that was there participating. Okay. The last thing I want to do here is acknowledge a bunch of people, and I have thousands and thousands of people, and I feel bad partly in doing this because I don't want to leave anybody out, but I do want to make some acknowledgments here. Dick Spohr and Earl Carpenter that I mentioned before, this is the Academy Awards part of this thing, by the way. I want to thank my dog and my, right? They're much more important than my dog. But Dick Spohr, Earl Carpenter, 2 teachers from Unity High School, E.C. Nelson, Bruce Halley from the lab in Stillwater, George Pearson, my PhD mentor here at Oregon State. I owe an incredible debt to George and to E.C. and to Bruce in particular. Chris Matthews, whom I've acknowledged before, is our department chairman. Andy Karplus, Gary Merrill, 2 chairs of our department who were instrumental in helping me to be successful doing what I do. Also wanted to thank Fred Horn, the Dean of the College of Science at the time I started teaching, who was very helpful, and also Sastry Pantula, recent Dean of the college, who also helped me career-wise, as well as all of my biochemistry, biophysics faculty colleagues. It's been a wonderful group of people to work with. I've had numerous students over the years. I'm not going to go through over each one of those because I feel I would be well, A, emotional probably, but B, would be not acknowledging other students. But I did want to put the names of quite a few students up here. And I could put hundreds, I could put thousands of names up here, truly. And I have been absolutely blessed with the most amazing students to work with, both in this class and as advisees. And one of the things that I will, and when I talk to other people about this, that I put in a very strong word for is the value of knowing, advising, in doing your teaching. And I've been able to put those 2 together, and because I have advised so many students and got to know students at that level, I feel that I've had a much better appreciation of the needs, the fears, the concerns, the aspirations of students in my classes as well. And so I strongly recommend that. And as some of you who want to be professors and go out and do some of the things like what I have done, I really encourage you to put your feet in both of those pools of water, because I think you'll find that very, very beneficial. The last 2 people on there, Neil Gladstone, relative to music, and of course my wife, Indira, who's made everything possible. Okay, now what I want to do next is do some singing, which of course, I don't want to have this all be serious. Some singing that we will do. And I had a request from somebody in the class who saw this song and says, I'd really like to be able to do it. And I don't usually do it because it's a long song. So I hope you'll indulge me here and join me in singing this song. It's fairly easy to sing. I'm going to bring up the, let's see, which one is this? Oh, no, let's see. That is not it. That's not it. The names aren't on here. I didn't realize. Oh, there's the name right there. Okay. That's not it. This is the one. Central. Okay, this one right here. All right. So please join me in singing this guy. Sung by Tim Karplus, by the way. Time a cell decided. Please join me. The time was right for it to split in two. Had to copy cellular instructions, for the daughter cell would need them too. Bring in a helicase up the DNAs to ease the stress. A gyrase joins the fray. Strands must be held apart. SSBs do their part, and primase builds a primer RNA. The clamp comes in behind clamp loader. DNTP's floating all around. In the wings, a replicase is waiting for the chance to start another round. Polymerase, my friend, starts at the 3' end. It puts a T across from every A G across from C. Perfect simplicity. The leading strand is made in just this way. Lagging strand is made in little pieces. Okazaki fragments you recall. Pol 1 fills the gaps that lie between them. Ligase comes in next and joins them all. Blueprints can't have mistakes, that's why polymerase corrects its work with exonuclease. Proofreading one by one till all its work is done. Hooray for DNA polymerase. An organism's cellular construction. Blueprints for the things they have to do. You can clap. Requires converting DNA instructions to ribopolymers. Oh yes, it's true. Because they've been bestowed with a genetic code. The RNAs provide the cell with means to link amino ways in most directed ways inside the protein making cell machines. It's a long one, I told you. Coli cells don't have galactosidase. And lactose should appear inside its food. The lac repressor leaves the operator, 'cause otherwise metabolism's screwed. Polymerase unwinds the DNAs it binds adjacent to the start site where it docks. Unravels A's and T's with such amazing ease at the promoter's little Tata box. Process moves along without much trouble, making RNA inside the cell. It all occurs inside transcription bubbles. Our bases get linked anti-parallel. mRNA then roams to find some ribosomes, subunits large and small by near the end. The AUG's in place inside the P site space. Initiation you can comprehend. We're almost there. Mechanism shifts to elongation, proceeding by 3 bases at a stretch. A GTP's required for translocation, advancing 5 to 3, the whole complex. Now everybody. The process moves on until a stop codon arrives and moves. I can't even sing my own song. Translation has to cease. A peptide gets released, and we have reached the central dogma's end. All right. Woo. Thank you. Thank you. This one's too long. We're not going to do this one, but if you, this song I'm actually kind of proud of. This song, there was a contest in Europe for science songs, and this was voted one of the top 10 science songs of all time. I was very, very proud of that. So I'm not going to sing that here. Okay. Now, let's see here. Sorry. What did I just do? Let's pull up. I've got one I have never done in class. You'll see why in a second, but I've never done it in class, and it's where's my bookmarks? There we are. You guys are going to be the first ones to do it, and let's find it here. Find BB. Okay, and here it goes. It's called BB, You're the Shits. It's not a bad song, but it's fun. Let me download it, and we will. Eric Hill's a good buddy of mine in the Honors College, and he and Heather Boren recorded this song. And Eric doesn't know I'm acknowledging him here today. He'll probably have words with me. BB, You're the Shits. Ready, set. This is a little hard to sing to. I'll let them sing it. The Green Acres theme. Oops. 50 is the course for me, with all its biochemistry. I soak up what this course provides, structure and function and info about peptides. 451's the choice for me, molecular biology. I love the way it gets risque, starting the bases inside of the DNA. Enzymes, 5 primes. Histones, your shits. All right. Okay. I've got one more song I want to do. This is the way I traditionally close this class. It's a clap along, okay? Now the problem with the clap along is people start going faster and faster as we go along, so try not to do that. But I usually clap. I can't clap with you with this selfie, with this selfie stick, so I'm going to get you started, and it goes. It's an old John Denver song. Keep going, I'll get you there. And don't speed up now. Okay, here we go. Ready? Get set. There's a bundle of things a student ought to know. And your talk is really very slow. Learning ain't easy. The lectures kind of blow. Thank God there's a video. We'll be gone through the cycles and their enzymes too. Studying the regulation. Everything is new. Gotta admit that I haven't got a clue. What am I gonna do? So I got me a note card, bought me a Stryer, got the enzymes down and the names he requires. Hope I can muster up a little more desire. Thank God there's a video. Just got up to speed about the NAD. Protons moving through the complex D. Electrons dance in the cytochrome C. Gotta hear the MP3. Fatty acid oxidation makes the acetyl-CoA inside the intermediates of the mitochondria. Very complicated, I guess I gotta say. Thank God there's a video. So I got me a note card, bought me a Stryer, got the enzymes down and the names he requires. Hope I can muster up a little more desire. Thank God there's a video. Replication kind of easy in a simple kind of way. Copying the bases in the plasmid DNA. G's goes with C's and T's go with A's. Thanks to polymerase. And the DNA is a template for the RNA. Helices and winding at TA. Termination happens and the enzyme goes away. Don't forget the poly A. So I got me a note card, bought me a Stryer, got the enzymes down and the name he requires. Think I could muster up a little more desire. Thank God there's a video. Woo. And that, folks, is the end. So thank you. Thank you from the bottom of my heart. Absolutely. Thank you.